Welcome to your 3-minute crash course on all things oncogenes. We'll cover history, definition, roles in cancer, and examples. Buckle up!
In order for us to understand what an oncogene is, we first need to understand its history. The first discovered oncogene was in the late 1970s by researchers J. Michael Bishop and Harold Elliott Varmus. They were expanding the experimental work of scientists like Deborah Spector, who researched animal virology and its relations to human medicine. Bishop primarily focused his study on retrovirity, a virus that infects and attacks a host cell by inserting a copy of its RNA genome into the DNA of a cell, changing the genome and causing oncogenes. He was inspired to broaden his oncogene research and concentrate on predecessors, also known as proto-oncogenes.
What is an oncogene? Oxford Dictionaries describes oncogenes as a gene which in certain circumstances can transform a cell into a tumor cell. But what does that mean? Let me introduce you to my friend oncogene, OG for short. OG's job is to code for the proteins. The code that OG writes allows proteins to direct cell growth. Before OG got the job title of oncogene, he was actually a proto-oncogene. While OG was a proto-oncogene, he was in charge of coding for proteins that help regulate cell growth and cell differentiation. Now you must be wondering, how did OG get a promotion like that? Well, that's due to one of 3 mechanisms. The 3 mechanisms are mutation, gene amplification, and chromosome rearrangements.
What is an oncogene's role in cancer? Let's think of a cell as a car. For a car to function properly, there must be a way to control the speed of the car. A proto-oncogene is like the gas pedal that helps to regulate cell growth and cell differentiation. On the other hand, oncogene can be explained as a broken gas pedal which causes uncontrolled cell proliferation. The unregulated growth of cells begins to outnumber normal surrounding cells, and it ends up forming a tumor. Let's look at an example. In the case of mitogenic signaling, which is the signaling process that promotes cell growth and division, a lot of proteins in the pathway can be oncogenes. If there is a mutation in any of the EGFR receptors, RAS genes, MAP kinase genes, or MYC genes, it would cause a dysregulation of the mitogenic signaling, which would lead to developing a cancer.
In this next section, we're going to go through some examples of oncogenes. The first is the HER2 oncogene, which is prevalent in breast cancers. The HER2 gene is used to control proteins and also acts as a networking receptor on the surface of cells that help them replicate. Therefore, if the HER2 gene mutates, it will expedite the growth of cells, thus leading to a tumor. Secondly, the KRAS gene is also involved in a number of cancers, most notably in pancreatic, colorectal, and lung cancers. When the KRAS gene is mutated, it tends to turn normal proto-oncogenes to oncogenes, meaning there will be an increased growth of cells, which also results in making it easier for cancer to spread throughout the body.